So today we're going to do an evaluation of an identification of a novel force-generating protein kinesin involved in microtubule-based motility, Vale et al. Here's our overview. So it was known by 1984 that cells move their organelles around in their cytoplasm, but not everything was known about their movement and not everything had been discovered yet. Squid axon was used to study organelle movement. In 1980, neurons, because of their bidirectional transport, were the basis of fast axonal transport, and therefore certain axons, if they were large enough, were great cells to study organelle movement in. Microtubules support organelle movement in non-neuronal cells. Microtubules were also known at this time to support organelle transport, so that was just another thing that they knew. Directed organelle movement within cells relies on ATP. It is assumed that movement is powered by ATPase because of this knowledge. All organelles move at the same velocity along microtubules. Since all of these do move at the same velocity, the same type of molecular motor or translocator may interact with a variety of organelles. Proteins that may comprise of the translocator are not known yet. Dynein is the only molecule that interacts with microtubules to generate movement. In 1985, dynein was the only known molecule to do this. However, no success has been made in finding dynein in neural tissue. And in this paper, many assays are used in order to biochemically identify the proteins involved in organelle movement. Okay, so for our methods. As I mentioned in the slide before, many assays were involved in order to determine what proteins are involved in organelle movement. So here we have the preparation of microtubules was done. Taxol polymerized microtubules, which were essentially free of microtubule-associated proteins or MAPs. They were prepared from both bovine and squid optic lobes. And the ones from the squid optic lobes were sometimes in conjunction with the preparation of the squid translocator. A microtubule bead and organelle movement assay was done. In order to measure microtubule movement, a suspension of microtubules, which were purified, were combined with a test sample on a glass cover slip. In some cases, the microtubule pellet was directly assayed for movement by placing a specific amount of microtubule directly on the cover slip in an ATP-containing buffer. This microtubule movement was visualized with a video microscope, and a sample was labeled positive if microtubule movement was present and negative if it was not. For determining movement of beads, glass cover slips were treated overnight with a solution of poly-D-lysine. This basically prevented microtubules from moving across the glass, so you could focus on just watching the beads move along the microtubules. A small amount of this mixture was then combined with microtubules on one of the coated cover slips, and the movement of beads along the microtubules was evaluated and observed. The organelle movement assay was done by combining microtubules with organelles in a test sample, and a 2 to 5 about minute video was recorded of a specific field of these microtubules to determine the number of different organelles moving per minute. An ATPase assay was done. The ATPase activity was measured according to the procedure that was given in the paper. Protein samples were combined with ATP containing 32p ATP and microfuge tubes at 23 degrees Celsius for up to 40 minutes. For the microtubule affinity purification, it was done with the purification of squid kinesin and the purification of bovine translocator. So for the squid optic lobes were prepared using homogenization, centrifugation, incubation, mixture with squid microtubules, and resuspension. The amount of 110 kD polypeptide that was present in the microtubule pellet and supernatant were determined through an assay by polyacrylamide gel electrophoresis. For the bovine purification, white matter was obtained from brains of freshly killed cows and homogenized, centrifuged, and mixed with bovine microtubules, resuspended, and incubated. The supernatant containing the bovine translocator was collected and then evaluated. For the polyacrylamide gel electrophoresis, this was performed in polyacrylamide gels under denaturing and reducing conditions, and then it was stained with Kumasi blue. Okay, so for our figures and results, this is figure 1. It shows the results of the co-sedimentation of soluble axoplasmic translocator with purified microtubules. Previous studies that were done suggested that the translocator does not bind tightly to the microtubules. This was confirmed because no microtubule movement occurred when microtubules were treated with the S2 supernatant. However, the AMP-PMP induces attachment of exoplasmic organelles to microtubules. So the thought was that if the attachment of organelles was due to an increased affinity of the soluble translocator for the microtubules, it might be possible to bind the translocator to the microtubules using the AMP-PMP. So addition of the AMP-PMP to purified microtubules and axoplasmic supernatant actually greatly increased the amount of a polypeptide that was present in the pellet with the microtubules. This band often appeared as a doublet, possibly indicating that there are two forms of the protein that differ slightly in charge or molecular weight. Two prominent proteins with molecular weights higher than 300 kD also associated with microtubules, but the amount bound was not influenced by the AMP-PMP. That can be seen in rows D and F, but D was the only row to show positive above it, which indicates that microtubule movement occurred. Proteins released from microtubules treated with the S2 supernatant in the absence, so that would be lanes C and E, they did not have AMP-PMP. They included the 300 kD proteins, but not the 110 kD protein. They did not induce microtubule movement. Lane D, the one that did show up positive, showed proteins released by ATP from AMP-PMP. Treated microtubules showed a prominent band at 110 kD and also did induce microtubule movement. Okay, so now for figures 2 and 3. So for figure 2, this shows the results from co-sedimentation of a translocator from squid optic lobes with purified microtubules in the presence of AMP-PMP. This is kind of similar to the slide before, but instead this was performed using a higher speed supernatant, or S3, from the squid optic lobes in order to obtain larger quantities of the translocator. So as you can see in rows F and H, when S3 supernatant from optic lobes was combined with the microtubules and the AMP-PMP, a polypeptide of 110 kD co-pelleted with the microtubules. So in this row, you can see around here, we do have those bands at 110, but in all the other ones, you don't see those. This, as I said, this prominent band was not seen in the absence of the PNP treatment, which basically you can see in all the other samples. Unlike the S2 supernatant from axoplasm, S3 supernatant from optic lobes did not induce microtubule movement. So you can see in row A, you have the S3, and this did not show movement. This could be either because the concentration of the translocator was too low or because inhibitory proteins were present. Proteins released from microtubules not treated previously with AMP-PMP, which included HMW1 and HMW2, but not the 110 kD polypeptide, did not induce microtubule movement. This can be seen in lanes C and D. Okay, and on to figure 3. This shows ATP and KCl dependent release of the translocator from microtubules. Each lane D through K, with D having nothing in it, but the rest of them have a specific amount of ATP and KCl mixed with the S3 supernatant and the AMP PNP. Lane A shows the 110 kD polypeptide and HMW1, it shows, that they remain attached in the presence of the ATP or KCl. This means that the 110 kD polypeptide could be increased without the resuspension of the microtubules to release the HMW2 and other proteins. When the 110 kD polypeptide was released with KCl or ATP, it did show between 15% and 45% of the released proteins. This helped to determine that assuming that 100% association of the translocator with the microtubules occurred with the presence of AMP PNP, the concentration of translocator in the S3 supernatant was estimated to be approximately 5 to 10 micrograms per milliliter. The 110 kD polypeptide dissociated from microtubules in either 0.1 to 0.3 molar KCl or 1 to 10 millimolar ATP. That is what is in lanes D through K. ATP was a more effective releasing agent than KCl, and combinations of the ATP and KCl did not produce a greater effect than just the ATP alone. KCl also released proteins that remained bound when only ATP was added. And the HMW1 protein was also released from the microtubules by ATP, as were the 65 or 70 kD doublet and 80 kD polypeptides. Again, you can see these in lanes D through K, but really E through K, which are all positive and do show microtubule movement. Okay, so figure 4 shows the gel filtration chromatography of squid translocator. Supernatant containing the 110 kD and the 65-70 doublet polypeptides released from the microtubules promoted microtubule movement. In order to determine whether these polypeptides actually induced movement, the microtubule purified translocator was further purified through chromatography. As you can see in figure 4A here, the major peak of ATPase activity from the gel filtration column corresponded to the HMW1 protein. While there was little activity that eluted with fractions that induced microtubule movement and contained the 110 kD polypeptide. The ATPase activities in the fractions corresponding to the peak elutions were respectively 0.13 and 0.01 millimoles per minute per milligram. And in the 110 kD peak fractions, the ATPase was activated by Triton or KCl, but it was not significantly inhibited by the NEM. The ATPase activity of the HMW1 peak, on the other hand, was inhibited by NEM. However, microtubule movement was not inhibited. Because microtubule movement occurred under conditions in which the HMW1 ATPase was inhibited, this ATPase was assumed to be not involved in powering microtubule movement. Okay, as we can see in figure C, the polypeptide compositions of fractions 21 through 45 were analyzed by the SBS page fraction containing so fraction 30 contains, which is right here, contains approximately 65 micrograms per milliliter of protein. The microtubule translocator eluted from the gel filtration column as a single peak. You can see that here. That corresponded precisely to the elution peak of the 110 kD polypeptide as determined by the SDS gel electrophoresis. So if we look at the gel now, the gel shows that the fraction 30 had the greatest movement-inducing activity from any other column. That can also be seen in C that I showed. The tubulin doublet at 55 kD that streaks across the gel here, I just thought I'd mention it because they mentioned it in the paper. It streaks. It is not typical, but is an artifact of this particular gel. The 65/70 kD doublet, which also demonstrated AMP, PNP dependent binding to the microtubules, as we saw in figure 2 earlier, it did travel on the gel with the 110 kD peak. This indicates that these polypeptides are tightly associated. The translocator purified by gel filtration chromatography induced microtubule movement at a total protein concentration as low as 5 micrograms per milliliter and at ATP concentrations of 10 to 20 micromolar. Microtubule movement induced by purified squid translocator was abolished by vanadate and was only slightly inhibited by a lower concentration of it. And that's figure 4. Move on. We missed figure 5. Figure 5 is showing the hydroxyapatite chromatography of squid translocation purified by gel filtration. Samples corresponding to fractions 28 through 32 from the gel filtration column shown in week 4 were pooled together and then applied to this column. Microtubule movement inducing activity correlated with the presence of the 110 and 65 kD polypeptides, and fraction 40, as you can see right here, contain the most protein at approximately 35 micrograms per milliliter. Microtubule purified translocator could be further purified by another type of chromatography, movement-inducing activity, as well as the 110 kD and 65/70 kD doublet polypeptides bound to this column were eluted together by KCl, and this indicated that these polypeptides are involved in microtubule movement. So figure 6 and 7, these show the purification of a translocator from the bovine brain. Figure 6 shows the co-sedimentation of the bovine brain polypeptide with squid microtubules dependent upon AMP and PNP. So the bovine brain S3 supernane was incubated with squid optic lobe in the absence, which is shown in lane A, of AMP PNP, and the presence, which is shown in lane B, of the AMP PNP. S3 supernane from bovine brain did not induce movement of microtubules by itself. When incubated with squid microtubules, MAP1 and MAP2 co-pelleted with the microtubules. And also when AMP and PNP was added, another major polypeptide of 120 kD co-pelleted with the microtubules. This polypeptide attached with equal affinity to the squid and bovine microtubules. And this and a minor polypeptide of around 62 kD were the only ones that demonstrated AMP, PNP dependent binding to the microtubules. However, there was no movement of microtubules that was seen associated with these proteins. Even when ATP was added to the 120 kD polypeptide and added to new microtubules, the released protein still did not induce microtubule movement on glass or in solution. Figure 7, however, shows gel filtration chromatography of the purified bovine translocator. Figure 7a shows the microtubule purified bovine proteins run on a biogel and their yields for the profile of ATPase and movement-inducing activity shown. Figure 7b shows the polypeptide compositions of fractions 21 through 37 analyzed by SCS Page. And figure 7c shows a densidometer scan of the Kumasi stain gel, specifically lane 30, which had the greatest movement-inducing activity. When the MT purified and ATPase released 120 kD polypeptide from bovine brain was run in the biogel column, the 120 polypeptide eluted in the same position as the 110 kD polypeptide from the squid. It also co-eluted the 120 kD polypeptide with the 62 kD polypeptide. They appeared as single bands rather than doublets, however, in this particular gel. Okay, on to the results. So we have a table here that I chose to put in the results because I kind of think it showed the different bead movements and kind of wrapped it all up. So the supernatant from axoplasm S2 promoted linear ATP-dependent movement of purified microtubules at 0.4 micrometers a second along a glass cover slip. In the presence of this supernatant, microtubules in solution also moved relative to one another to form a contracted aggregate of microtubules. Carboxylated latex beads, when they were treated with S2 supernatant, also moved at the same speed that the microtubules moved. And the microtubules or beads incubated with ATP without the S2 supernatant did not exhibit directed movement. The number of organelles moving along microtubules was greatly increased by the addition of the S2 supernatant. And then to talk about the movements with the squid and bovine translocators, the squid and bovine translocators were purified by microtubule affinity column and column chromatography, and assayed for their ability to move microtubules in a solution along glass as described above. The velocity of microtubule movement on the glass was 0.3 to 0.5 micrometers per second, whether induced by purified squid or bovine translocators or the S2 supernatant. The column purified squid and bovine translocators also moved beads along microtubules at the same speed. This suggests that bead movement along microtubules is unidirectional in the presence of purified translocators and that they depend on activities of the same protein. The squid translocator purified increased the number of organelles also moving along purified microtubules immensely compared to the number that moved in the ATP-containing buffer alone. The purified bovine translocator, on the other hand, did not consistently increase the frequency of movement of organelles isolated from the squid axoplasm. Organelle movement induced by the S2 supernatant or purified translocator appeared to not depend on nonspecific association of the translocator with the organelle surface because of multiple organelles that did not exhibit directed movement even when the active translocator was present. This experiment suggests that the translocator is a protein that binds reversibly to organelles into microtubules. For this new molecule that they did discover, they proposed the name kinesin. Okay, now our discussion. So microtubule movement occurs only in the presence of the major polypeptide with a molecular weight of 110 to 120 kD and with less abundant polypeptides of like 65, 70, and 80. Binding of these polypeptides to microtubules is definitely enhanced by AMP, PNP, and is diminished by ATP, conditions that correlate with induction of microtubule movement. It is believed that the 110 kD squid polypeptide and the corresponding 120 kD bovine polypeptides are homologous proteins due to their shared ability to induce movement of microtubules and carboxylated beads. Among a few other similarities between them, the translocators isolated from the squid and bovine tissue are not dyneins, though. This is due to several tests. It is proposed that the translocators be called kinesin, and kinesin has many possible roles in biology that still need to be researched further. Possible roles for kinesin are they appear to be a binding site for a structure to be moved. They appear to also have another site capable of cyclic on and off interactions with microtubules. They also can act as motors for organelles. The results from this study indicate that kinesin powers organelle translocation along microtubules. However, evidence is circumstantial, so many more studies need to be done in order to confirm this. It is also known or predicted that column purified kinesin can increase the number of organelles moving along purified microtubules, and that it induces beads to move along microtubules, as we found from this study. On the basis of all of these findings, it seems likely that kinesin binds to organelles and moves them along microtubules, although there is no direct proof of an interaction of kinesin with organelles. Thank you.